You are welcome to part 4 of the module 4, where we are going to see the 16S ribosomal RNA bioinformatic processing pipelines. In this session, I will show you different pipelines that are used to process microbiome data, and at the end of this, I would expect you to be able to decide which pipeline you can use for your own data processing, or which pipeline you need really to invest more time to understand it. So the first pipeline has been developed by H3 Bionet Africa. It's based at the Computational Biology Division at the University of Cape Town. And it has three phases, three major phases. The phase 1 is the pre-processing of the reads. And the second phase is the auto-picking, classification, and phylogenetic tree generation. And the last phase is the measure of diversity and the other statistical parameters. So it's very important that all these phases are clearly executed when you are processing your data to ensure that the final results are really representative of what is found in your data set. So the first phase is the pre-processing steps. In this phase, first of all, you have to do the demultiplexing of your sample if they are not yet demultiplexed. Demultiplexing is the process whereby you assign each sequences to our respective samples. This is very important because in high throughput data, when we do the sequence, we pull several samples together and you sequence all of them once. But we are able to assign each sequence to the appropriate samples because when running the PCR, the reverse primer had a barcode, which is a unique identifier for each sequence based on their sample. So we do the demultiplexing to assign each sequence to their respective sample before we do the further processing. Then we have to check for short read lengths using FastQC. As I showed earlier, we see the short reads. These must be checked out and removed from the data set. You have also to remove the low quality sequences because these they compromise the final result. The quality of the final result will be poor. But also we have to remove the adapters and the primers. This is very critical also because if you don't remove the adapter and the primer, you may think you have come up or discovered a new sequence in your data set, but actually this is because of the adapters and the primer sequence, which is still part of your data set. But also we have to remove the chimeric sequences. These are the hybrid sequences which normally happen during the PCR steps because when you run the elongation steps, you may find the first part of the cycle then merges with the second part of the cycle, then you get the hybrid sequences. And if you don't remove these, also you may think you've discovered a new bacteria or new types of microbes, but actually this is because of these chimeric sequences. These must be removed in your data set before you do the further processing. Then you have to merge the reads. This is very important because by merging the forward and reverse read, actually you increase the quality of your sequences. There might be a position within your sequence which is having the poor quality for the forward or the reverse read individually, but once you merge them, then you improve the quality of the consensus sequence. This is very important, and we take advantage of this when we are using the high throughput data, especially from Illumina dataset. The second part is the auto-picking, and here we want to assign our sequences to operational taxonomic unit. Okay, so an operational taxonomic unit is an operational definition of a species. Auto defines a species or a group of species often used when only DNA sequence data is available. So you can use a sequence data to assign the operational taxonomic unit of a given community. So we have to pick here the auto-picking. So the auto or the cluster of sequences are clustered based on their similarity or their identities. So there are three different steps where can be used for auto-picking. There is so-called de novo auto-picking. This is whereby you assign sequences to autos based on their similarities to each other, and you take the similarities at around 1 to 3 bases difference. If they are similar at more than 97%, then you put that one into one auto. So you cluster sequence without basing on the reference sequence. But again, you can use a closed reference auto-picking. In this case, you take your sequences, and you use the reference database of the already known sequences to assign the operational taxonomic unit. In this case, the closed reference, you may come up with samples or with sequences that are not present in the database, so you will miss those assignment to those sequences if they are not already reported in the database. The third approach is using open reference picking. In this case, you use your sequence, your assigned sequences O2s based on the reference, but for those that are not present in the database, then you can cluster them together to create another O2. So any of these three approaches can be used. It depends on the interest of the researcher to choose which approach to cluster the sequence interpretation to Superman unit. Then a representative sequence is selected for downstream analysis. Why a representative sequence? We want to minimize computational demand, so we take only the representative for further analysis after doing the auto picking. But remember here, we will also at this stage we will also be having the O2 table where we know exactly the frequency of each O2s in the dataset. How many times does it appear? But for the further processing, we continue with a representative sequence. So the third phase is taxonomic is a classification steps. At this stage, a taxonomic identity is assigned to each representative sequence. We assign each sequence to its taxonomy. Classification are done against the three main reference database by aligning, so the sequences are aligned to the reference database, and the threshold of similarity is set to decide whether this sequence, if it matches at this percentage, then you judge it as a particular taxonomy. So there are many types of database that are used for that case, but they must be the validated one, because if they are not validated, then they may not represent a true taxa. So we have the Green database, for instance. We have the ribosomal database project. Also, we have the Silver database. They must be the curated one, so that when you assign your sequences to them, they are true representation of what you found in your data set. After the classification, then we do the alignment. Alignment is the first step in the generation of phylogenetic tree to understand the evolutionary relationship between the samples. Remember when I was introducing the 16S gene, I said because of the conserved and the variable region, we can get the evolutionary history of those microbes present in a given area. So we must do the alignment with the reference database, of course, to see what could be the relationship between the organisms that are found in a given area. So the aligner of choice are those that does alignment to a template or secondary structure of the 16S sequence. So there will be a template, already curated, annotated template will be used to assign, to align our sequences. After the alignment, then you create the phylogenetic tree. The phylogenetic tree represents a relationship between the sequence in terms of the evolutionary distance from a common ancestor. Those that are very close, they will be very closer in the phylogenetic tree, and those that are very far, then they'll be equally distantly separated from each other. In downstream analysis, this tree is used to example calculating the unifrac distance and some alpha diversity measures. There are several statistics that can be done using the tree to see the connection or the link between those microbes found in a given area. So this tree is of paramount importance for the downstream steps. So there are other pipelines apart from the H3 developed H3 BioNet developed pipeline. We have other pipelines. We have the so-called Mothur pipelines. The Mothur pipeline is also commonly used. There is a big community working on these types of pipeline. It has been developed by Patrick and the group in the US. It's a community developed platform, and there is a very serious forum whereby if you are stuck, you can ask your question, and these guys will be there to assist you or to guide you on how to use the platform. So I've personally also used this platform for analyzing my data when I'm doing my PhD, and I've summarized some of the steps that are used in this pipeline. And they've generated a very good standard operation procedure, which you can easily follow. Even if you have not been a mathematician, you can follow the standard operation procedure to be able to use all the steps that are needed in this pipeline. So the summary that I've come up about is like you have the raw sequence read, then you have to do the quality control using the Mothur tools. Then after the quality control, in the quality control part of it, you make the config where you filter those poor quality sequences or poor nucleotide sequences. Then you align with the reference database, in this case is Silver. Then you remove the chimeric sequences to ensure that you have a true representation of your data set. After that, then you assign to the taxonomy, or you assign to the OTUs. Again, using cysteine ribosomal database as a reference, you cluster the sequence at a 97% sequence similarities. And then after taxonomy assignment, then you can do the alpha and beta diversity analysis using several index that are available or several metrics that are available, and all these have been clearly put in the standard operation procedure. I believe everyone in this group can easily follow if you are keen to see how this tool works. Then after the abundance and diversity analysis, you can do a further statistical analysis. After generating the biome, you can do after generating the biome and the metadata of the dataset or of the output you get, then you can use for further statistical analysis using the R package or any other tools that are used for microbiome research. So basically, it generates the phylogenetic and generates also the biome file. From these two output, you can do other analysis. And if you can follow online in the forum, there are several steps or several approaches that are used to analyze this data set. I'll give you a link later where you can follow to do if you want to practice on how this Mothur package is being used. Another mostly used pipeline is the CHIME pipeline. This is a big community used pipeline, and it has been developed by Caprasso Group, Gregory Caprasso Group, also based in the US, and it's a community-based tools, open source, you can freely use, download and install in your machine and use for the data analysis. And again, they have the very good forum where they can exchange ideas or make a comment or raise any query or get any suggestion. So if you are stuck, then you are assured of getting help. So what is CHIME? Is a quantitative insight into microbial ecology. It's a next generation microbiome bioinformatics platform that is extensible, free, open source, and community developed. It is a powerful suite of microbiome and analysis package where researchers can start an analysis with raw DNA sequences and finish with publication quality figures and statistical results. This is very important. You start from raw to the quality that's good for publication, with all statistics, with all the graphs, everything you need for the publication. So it's a very super tools that is also available for microbiome research. It's a complete design. We had the CHIME 1, now we have CHIME 2. Is a complete design of CHIME 1, which still retained features that makes a powerful and widely used tool. So the latest version is the one which is mostly used, and they are really encouraging to use the latest version as opposed to the older one. So the key features in this tool is that they have a tracking and exploration mechanism. For instance, you can once you do the analysis, you can clearly check all the steps that you have used and refer to all citations that have been used in that steps. But again, you can interactively check the status of your output, explore the status of the output that you generated. Like you get the figure, then you can explore the figures to see what really they mean. Again, you can easily share your output with your collaborator, even if they don't have the tool installed in their machine. They can contribute and see what's happening. And again, the other advantage is that it's a suite of tools. There are several packages within these tools, so that you have those they call plugins based systems. You have different plugs for different objective or for different statistics, which is very, very good for the analysis. So this is really a great tool for the microbiome work. So this is a conceptual workflow. I would like to start this in the second session. Let me end here. I'll proceed with you after a while, because I wish you to understand all these steps, because they are very important, why we are doing it and how do we do it. So I'll explain in the next session of our meeting. Thank you.